So welcome to video 2 in our course on exploratory data analysis. Here we're talking about statistical testing on microbiome data, and that's the focus on our this particular video. Later we'll talk about dimension reduction, feature selection, and feature extraction before discussing principal components analysis as a key method in high dimensional data analysis that is complemented by multidimensional scaling as another key method. And we'll also talk about two other techniques, TSNE and UMAP, that are also relevant for trying to interpret biological data. So let's look at microbiome data first. So we get the example in the previous one where we had different centers doing different microbiome sequencing, and we looked at percentile normalization as an example of that. So what we want to do this, apply this method more directly, is that within each project or study, get the controls and then map the cases on top of that. So we want to get the percentile for each case as if it was mapped onto the distribution of controls, since that's where the percentile comes from we're using the cases as our proxy and considering across the distribution where our cases land. So for example, if you human heights, you might find that someone with a height of 2 meters has a very small percentile, maybe they're at the top 2% of heights, whereas someone who's a height of 1.5 meters might have quite a large percentile because they're smaller than average. So we're extracting those percentiles from the data. And this would be for a particular taxon. So we cross different taxa, and therefore you would get a lot of data at the end, obviously, from all of this. And you could repeat this across all taxa and across all studies. And this means that we can compare from this data, this data then between studies much more effectively. An example of this, we want to look at a particular species such as Bacteroides fragilis and look at that abundance after we do our transformation. It could be using CLR transformation or some sort of simple relevant relative abundance. It depends on our approach. And what we can find if we do this is that the fraction of p-values that are small for the raw data is very, very high because there's a huge amount here of false positives. If we do the percentile normalization, our expected value of rate of false positives appears to be low because our fraction of small p-values is quite low. What we can see there, if you look at combat normalization or limit-based normalization, which can be good normalization methods in the right way, in the right context, we find that they don't perform quite as well. So therefore, we need to think about for any particular data set how we normalize it that's specific to that data. We can't just apply any technique and check that it works effectively. So this is an example of how percentile normalization can reduce false positive rates by using the appropriate correction method on microbiome data. An illustration of this, and we'll talk about it more in the demo videos, is where we have two different studies. And what we have within these studies is that distribution of cases and controls using data for a particular ITU. And what we can see is, which you would expect, is that there's kind of a decay here as we go across the frequency of that particular OTU. And it's the same in the other study. Overall, there's the same pattern, but just the counts are differ, and the level of sequencing, if you're doing metagenomic sequencing, the depth of sequencing will differ, and therefore the count data will differ. So they are the same underlying distributions, but their shape are quite different. So we've got the controls here, and the cases are higher, but are they comparable? So what we can do is create as our controls become our index, and we map the cases on top of that. So what you can see with the cases is that the percentiles of where they're placed onto that distribution does match what's on the left because most of them are higher. And same for the study one up here. You get that same pattern, but the exact placement depends on what data we're looking at. And what we can do then is merge the data. So this is the key thing. If we don't normalize it in the right way, we can't merge the data, and we're comparing apples and oranges. So here we can now merge the data and therefore apply this kind of rank-based normalization before merging the data that we can now analyze together. So that's the kind of quick summary of how percentile normalization works. An example of this will be that we have a bunch of controls for a particular study, and we have two cases, and we consider then if the controls above are our distribution, where do our cases fall into the percentiles that might be present. So what we can do is take case 1 where the abundance is 0.06. So what we see when we sort these ones here is just 0.04 is more extreme than that. So therefore its percentile is 0.2 because it's this kind of 5 points along our scale. So that's where it falls. If we look at the second one, it's bigger than all of them except one at the end, 4 out of 5, so its percentile is 0.8. If we look at study 2, we have these different control abundances, so you can see that they're inherently higher, and if we can map these onto that distribution in the same way, where 0.09 is smaller than all of them, so its percentile is 0, 0.045 is kind of in the middle, well, it's toward the end, and therefore its percentile is 0.8. So that means we have these outputs from that. So we don't need the control data anymore. We just have the case data and we have the percentiles there. And then if we compare, if you imagine we have lots and lots of data, we could compare the distributions for those percentiles to each other in a more robust manner. So we kind of see that there, that if we had a large data set, that we would look at the relative abundances of the two different studies and be able to percentile normalize so that we can interpret them in the right way. So there's different ways that we'll talk about doing this, one of which will be the classic way will be down here, this Wilcoxon rank sum test, where we compare the ranks of the distributions to see if there's any difference between the distributions in terms of their mean or location and their variance, their shape. We'll also look at permutation testing, which is a way of resampling the rearranged labels on a collection of samples randomly until for whatever 1,000, 10,000 iterations, and looking at intrinsic inherent distribution in null data comparison and where our test data, where its statistic maps onto that particular null distribution. So it's kind of an empirical way of dealing with heterogeneous data. So this permutation test is a type of non-parametric test and looks at a null where the underlying distributions might be heterogeneous, and it kind of says, okay, our value of interest places at this line, and this is our null distribution here, and how extreme is that line? And that can be typically be based on the test statistic, but it can also be based on other features depending on what's of interest. And there's a few papers which apply that in different contexts that are listed here as well. So this permutation testing is a type of resampling without replacement. It considers what does the null look like, and can we resample the null, the controls to figure out their distribution. And therefore mapping the cases onto the controls distribution to see if the cases are extreme. And then we iterate through a large number of permutations. We rearrange the labels multiple times and can keep updating the statistical test to measure that. An example of this will be looking at alpacas, and here we're talking about shampoo, but we could have other questions like looking at antibodies for a coronavirus, for example. And what we're trying to think is, does a new treatment increase nanobody production, or does it clean the alpaca's hair more effectively? All these sort of questions we could ask. What we're going to do is assign half of the alpacas to a new treatment in red down here, and the other ones are going to keep the old treatment. What we can do is use R to make random numbers to measure these things and compare those different groups. And then we can measure this response. We would typically use a t statistic or an adjusted t statistic to measure these differences between the groups. So it's taking the difference in the means divided by the square root of the pooled standard deviation. That's the denominator here, where we have the group n is the one on the left. That's these ones here, and it's the standard deviation squared divided by the number in the group, which is 12, and same for the group in the right. And if the nanobody treatment is correct, is better, here for n, we'd expect that value to be higher. And therefore we can randomly permute this test and mix up the labels. So it's randomly going to be red and blue for each group, and continue that and repeat it many, many times to create an empirical distribution onto which we can map our particular value. So we can, if the value is here, then this area on the right gives us a sense of how extreme it is. And we just should simply count the number of instances that are on the right that tell us how extreme our value is. And you kind of see that there. We count up those instances, and here it's 16 out of 200. It kind of tells us that our p-value, our permuted p-value, would be p is 0.08. So this is quite a robust way of dealing with heterogeneous data that doesn't have an underlying distribution. So what we're saying then is that given our model and our null hypothesis, the chance of getting a statistic more extreme than the one that we observe is about 8%. So that kind of concludes this section. What we can ask is how to move on to next time. We'll look at dimension reduction and feature selection and feature extraction next.